Hello everyone, this is Richard from Modern Healthy Hong Kong. Today we have our first interview with Professor Moshe Szyf of McGill University, who has profound knowledge of epigenetics, DNA methylation and biological aging. We will be releasing the interview as a series, with each video focusing on a different topic. Today's topic is, what is epigenetics and how does it affect our lives? Our epigenetics is closely tied to the way that we age. An understanding of epigenetics is very helpful in giving us insight into why and how we age and how we might influence it. First, let me introduce Professor Szyf. Professor Szyf is a James McGill Professor of Pharmacology and Therapeutics at the McGill University, where he also holds a GlaxoSmithKline-Canadian Institute of Health Research Chair in Pharmacology. He is one of the pioneers in the field of epigenetics. His lab has proposed 3 decades ago that DNA methylation is a prime therapeutic target in cancer and other diseases, and has postulated and provided the first set of evidence that the social environment early in life can alter DNA methylation, launching the emerging field of social epigenetics. This was the subject of his 2016 TED Talk on how early life experiences is written into DNA. He is also the founder and CEO of HK Therapeutics, a Hong Kong company dedicated to making early detection of cancer using DNA methylation widely available. So our audience may not be that familiar with epigenetics. So could you take a few minutes to introduce epigenetics to our audience?
Sure. No, I like to think about our cells in our body as mini computers. And our body has to do many, many different tasks. And it has to do them at different times in response to different triggers. It's an extremely complex program that is needed for us to be functional and to go through our lives. And all of it is encoded in the DNA. The DNA is the operating system. The DNA is really the hard-wired element of the computer, like the board. And on that, an operating system is written. The operating system is in the sequence. The same way we have Windows, or we have iOS or Android. The operating system allows that DNA to be programmed. But then you need software. The operating system by itself, if you have a phone with only operating system, you can't do very much. So you need to code software into the operating system. And this is exactly what epigenetics does. So during the time that we develop in our mother's womb, and afterwards throughout life, but mainly through the period of gestation, software is written in our DNA. And that software is programming our DNA in different ways in different parts of our body, but also programs our DNA how to react to events that will happen later in life. So our DNA program must be anticipatory. That is, it's not enough to be functional before the moment we are born. We have to anticipate problems that we'll have down the line. And all of this has to be programmed. For example, we have to program an immune system that will react to infectious agents that don't even exist today. So the beauty of the programming is that it programs our DNA in space and time and context so that our DNA can function throughout life. And we learned about that around 160 years ago. And when Darwin came with the origins of species, the main emphasis was on the heritable material. But Darwin already realized that the heritable material by itself isn't sufficient. There must be some sort of interaction with the environment so that the heritable material, that Darwin did not know what it was. Neither did he know how the environment interacts with the DNA. But he realized that DNA has to change in order to cope with demands of the environment. Part of this change is going through natural selection, which is evolution. But the other part has to happen at much faster time scales. And later on, when genetics was integrated with Darwinian theory, we understood that the genes are coding the secrets of life, but there was still a question. If we have the same genes in every part of our body, how can they do so many different things? In the 40s of the previous century, a British scientist called Waddington has coined the term epigenetics, which he meant, yes, we inherit the genes. Yes, they only change slowly in evolution. And yes, life has much faster demands on us. So how could one DNA do all of this? It can't be just in the heritable material. And then he suggested that during embryogenesis, during the time an embryo is in his mother's womb, the DNA is programmed by something that is beyond genetics, which he called epigenetics. But he had no idea what this material was. The same way Darwin didn't know the genes exist, but he anticipated their existence. Waddington did not know what epigenetics is, but he noticed that there must be something else happening to DNA. But that was a profound idea, because it suggested that not everything is encoded in the genes and something else is happening to the genes. And that has many, many ramifications, as we might discuss later on. And then in the last 50 years, the nuts and bolts of how epigenetic works were unraveled. It's a very complex chemistry. It's an amazingly complex chemistry that is programming our DNA so that it can function in space and time and carry us through lives.
Right. Yeah. So can you tell me some of the, so you say it gets programmed. So what kind of things would program?
Right.
Set the program.
So as I said, originally, Waddington only knew that it gets programmed. He had no idea how it works.
Right.
Because as the science of chemistry and biochemistry developed, it was discovered that the DNA itself has additional marks on it. In the 40s as well, a scientist called Hotchkiss discovered that DNA has, in addition to the 4 bases, what he called a minor base. And he had no idea where it comes from. It couldn't be heritable because, as you know, DNA is heritable. So we have 4 letters in our DNA, and each letter corresponds to another letter, and that's how it's replicated and is inherited. So we get an exact copy of our father and mother DNA. But that could not be inherited, but it was still a part of our DNA. And later on, this minor base was discovered to be a methyl group. And methyl is a chemical, it's the smallest chemical moiety. And when it's attached to one of the bases of the DNA, you get what we call today DNA methylation. Chemically, it's a new base, but it is synthesized or is made in 2 different processes. One process is the copying of your father and mother genome, which is the copying of the genetic information. And then afterwards, another machinery goes on the DNA and adds those marks. In the end of the day, it's the same DNA, has 2 levels of information. The genetic information that came from your father and mother, and the methylation, which was made when you were a fetus in your mother's womb. We'll learn later that those methyl groups are not added just randomly, it's not just noise, but they are added in a highly organized way. That when the baby is born, each cell will have a different profile of how these methyl groups are added, hence programming. Because programming is based on a binary language, 0, 1. So if you just break it down, all you see is 0, 1. So what makes program a program is where you put the 0, 1s, the order by which you have the 0 and 1. It's the same with methyl groups. By themselves, they're very simple basic chemical. But because it's positioned at different places in the DNA, it can give different signals and different programs. So for example, we have 20-something thousand genes. But in each cell of our body, we only work with a certain amount of genes. And that gives us the diversity that you see. We have eyes, we have faces, we have muscle, we have hearts, we have lungs. So lung will have a set of specific genes that work. And heart will have a different set of genes that work. So by marking which genes work and which don't, you can decide this cell is going to be a heart muscle and this cell is going to be a lung cell. And so on and so forth. We probably have millions of different cell types, even though we know only a few of them. But probably there are no two cells that have exactly the same methylation program. And that heterogeneity of program creates all the vast possibilities. It's like having a phone with a million apps. So you can't just make a phone call, you can do millions of different things. And our DNA is like a phone with a million apps. And so this is one element of the coding. It's simple to explain, but it gets much more complicated. And the science of epigenetics is an extremely complex science. Because first, we learned that even these methyl group can be further chemically modified by other, what we call enzymes. This is the machinery that does the job in the cell. We still don't know what these other modifications mean, but they make the language more sophisticated. So if you think about, you can have one layer of program and on top of it have another layer of program in the same DNA by having different kinds of chemical marks. And then the DNA has to be packaged in the cell. The DNA is 12 meters long. Our cells are nanometers, an extremely engineering challenge. It's amazing how nature has solved that problem by packaging this large amount of information and hardware into a very small space. And that packaging, which originally we just thought this is just packaging material, actually defines which genes are open to business and which genes are closed to business. So we have, like when you have money in a bank, you have money that is liquid and money that is frozen for 2 or 3 years. So in every cell, we have everything. We can take any cell and do anything with it. And by the way, this is why cloning works, right? You can take a cell from anywhere in the body and make a full animal from it, because we keep our DNA in different parts. The one that we use now, the one that we want to use only when certain signals come, the part of the DNA that we never want to use unless we have to change the fate of the cell. And so this packaging defines as well which parts of the DNA are open to immediate business, which part of the DNA will open to business only in response to certain needs, and which parts will be kept, for long-term deposits, like, in case we need to recruit cells from one part of the body to work in another part of the body. And that process of packaging involves extremely complex biochemistry and maybe more than 1,000 different chemical reactions. And all of them have to be orchestrated in an exquisite way, because any mistake can cost us our ability to live. And we'll talk about mistakes maybe later, because they can cause disease. So the perfection of this process is really remarkable. If you just examine it, you cannot but marvel from the beauties of nature. So epigenetics is probably the most beautiful part of nature. By creating these immense, mountains have very limited possibilities, and rivers and even stars. They're very simple to study. Epigenetics is much more complicated than astrophysics, because the number of processes and the perfection by which all these processes have to be integrated is infinite. And therefore, every time we think that we understand it, we discover a complexity. And that's the process of science. So, We kind of understand something, and then we understand, oh, wow, it's more complicated than that. And we dig further in and we find. And that's the marvel of science. And the amazing thing is that this whole thing works. And, you know, whether it's a mouse or a human, by the end of gestation, their eyes, their hands, their legs, their hearts, and they all breathe and they all regulate it. And they know when to go to sleep and when to wake up, and all the cells respond to this. It's a marvelous thing. And of course, the biochemistry is extremely, extremely complicated.
Right. Yes. I mean, I agree with you. It is amazing that it all fits together in the right way.
Yeah.
So that's kind of one element of the epigenetics, which defines, you know, the different types of cells within your body.
Right.
But like it also it can also change your behavior.
Right. Right.
Yeah.
So this is the next fascinating revolution, right? So the first revolution was discovering that we have genetic material, and that genetic material changes at the time scale of evolution. That was the discovery of Darwin. The discovery of Mendel was that he discovered, you know, who does the job of inheritance, and then it was put together in DNA theory. And when the structure of DNA was discovered, it was an amazing day in the history of humankind, because now we could put evolution and genetics together in one package, in a grand hypothesis, and understand how inheritance works. Why, if our parents had blue eyes, we also have blue eyes, and why do we inherit these things from our parents? But it gave a misleading sense of determinism, that everything is there. That essentially, don't waste your time. You know, if your father and mother gave you a good heart, you can eat whatever you want, you can drink whatever you want, you can smoke whatever you want, and you will live long. And in many cases, this is indeed true. You know, we know about families who live very long and are doing all the wrong things. And in the family, you know, I once saw a picture of 5 generations of a family. Everybody was obese, everybody was smoking, everybody was spending his days, you know, drinking alcohol and never exercising. So a lot is defined by genetics. But the question, the idea was that everything almost is defined by genetics. So the role for environment, and environment in a broader sense. So when we talk about environment, we always think about global warming or lead in the walls. But environment is also composed of really 3 major components. One is indeed the physical world, which is the amount of light and temperature, the poisons we throw into the environment, etc. But there's also bacteria and other organisms. So we are not alone in this world. Every human has hundreds of species in his body, and of course, millions of species around him. And interaction with other species is extremely important for our lives. And then, of course, there is the social environment. Humans, as many animals, we are social animals. So we develop social interactions from cradle to grave. And so all of these are working. And they were ignored. So the genetic theory was so powerful, was so elegant, and was so amenable and tractable to experimentation. Because once it's all chemistry and physics, it's very easy to study. You know, what makes biology complicated is that we cannot reduce it always to chemistry and physics. But once the laws of chemistry and physics, as sophisticated as they are much simpler than the complexity of biology. And once biology was reduced to physics and chemistry, it gave a huge push to medical science and to medicine. And the tremendous revolution that we saw in medicine in the last, 100 years came from the discovery of DNA and the way it works. However, one thing was missed, which is what happens beyond that, beyond the program that is evolutionary inherited. By the way, epigenetics is also evolutionary inherited, right? Because a mouse will never be a human. A mouse will always grow through the epigenetic programs of a mouse. And every cell in his body will be in the same place. So that has to be encoded in the DNA. But what happens when we encounter other things? Do they change? And none of us thought about this. None of us, I'm a hardcore biochemist. I was trained as a biochemist and geneticist. We never thought about social or environmental events as something we need even to bother, except in the context of natural selection, which is too long for us to be bothered by. And so I encountered this question for the first time when, by some serendipity, I bumped into a neurobiologist, a psychologist who was interested in maternal care. And that was totally serendipitous. It happened in a bar in Madrid. And, one of the things, by the way, that social distancing does, it reduces serendipitous meetings, right? Because we have to plan so you can't bump into somebody in a bar. But actually the greatest ideas come from serendipitous encounters. And he just gave a lecture on maternal care and the impact it has on the physiology of a model that he was studying, which is rats. And it took a while for me, but it dawned upon me, how is it possible that the level of maternal care, which is completely social, it seems, it can affect physiological, kind of hardcore functions of the body. And it led us to start collaborating and actually putting together two lines of thought. The social sciences, psychology was thinking one way, the biologists were thinking another way. And in the last 20, 25 years, there's a synthesis, this next synthesis that is emerging, which is trying to understand whether these processes that sociologists call them social processes, are not also embedded in the same biology that we were always studying. And are they totally different? Or are they using exactly the same tools that biology is using? But these cannot be inherited in the simplistic way, because they are dependent on things that we don't anticipate. Is our mother going to treat us in a certain way or another? Or is it going to be a disaster, a natural disaster that will change everything? Are we going to move or immigrate? Are we going to be poor or rich? All these things are not anticipated. So how do they work? And there is a large body of data that comes from economics, environmental science, that suggests that all these things are important. They affect our health. They change, trajectories of health. So, for example, I came across information that I never knew, which is the relationship between socioeconomic status and physical health. So one can understand how socioeconomic status can affect your social behavior. But why would it affect your heart rate or your blood pressure or your insulin resistance or your asthma? So all of this has pushed the science and myself to start asking for mechanisms. And essentially, like the previous synthesis, like the discovery of epigenetics, we took the principles of the past and put them together, and we have now a new Gland synthesis, which is the same idea that operates in development, that different apps are coded into the genome, and that the potential of these apps is really infinite. If you have a smart coder, the apps can change. Why should it stop at birth? Maybe environment is writing apps in our genome. Maybe social environment is also writing apps in our genome. So this idea was considered impossible 15, 20 years ago. But again, once you delve into the biochemistry of it, you start building biochemical principles. Now it's again reduced to physics and chemistry, and you can start understanding it. Of course, not its full complexity, but you understand its potential feasibility, that it is possible that maternal care will have long-term physical consequences, because maternal care can program the DNA. And the question is, how can maternal care program the DNA? How can social distancing program your DNA and have consequences that are well beyond the immediate threat of a virus? And how a natural disaster that happens a short time in life can affect not just one generation, but multiple generations. And so I would not, say that we know everything about it. We know very little about how that works. It's still a mystery. It's still an enigma. But we have some ideas that suggest the feasibility of interaction between, ephemeral things like social environment and our DNA. And the maternal care model provided us with a tractable, scientific model where we can explore potential mechanisms. And we indeed found that the environment, the social environment early in life does program the DNA using exactly the same tools that development is using to program our DNA. So now we have a third element in our DNA. First is the genetics, what past generations have given us, our ancestry. Second is the epigenetics that was formed in the womb of our mothers. Again, both elements are highly deterministic. And then comes the freedom. The third element, which is environmental or behavioral epigenetics. The stuff we do that is not anticipated by our fathers or by our epigenetic process during development, but happens. Genetic identity, cellular identity, which defines which cells in our body do what, and experiential identity. And all really operate through the same biochemical mechanisms.
Thank you all for watching. I hope that you found the videos informative. We will release the rest of the videos soon, so please do subscribe and join us for the rest of the series. I wish you all well, and I will speak to you again soon.